Okay. Thank you for joining us to the second part of this session. For the last 10 years, the incorporation of immunotherapies into treatment regimens has been heavily investigated and has revolutionized the treatment of solid tumors. In the field of sarcomas, there are several challenges with immunotherapy use, as several studies have already shown immune checkpoint inhibitors tend to have limited efficacy compared to other solid tumors due to tumor heterogeneity. However, other studies tend to demonstrate that when combined with other therapies, immune checkpoint inhibitors may sometimes show some activity in certain subtypes of sarcomas. This raises a lot of questions regarding the role of immunooncology and T-cell therapy can have in the treatment of soft tissue sarcoma. And this is why I have the pleasure to welcome our longtime friend, Dr. Jon Trent, from the Sylvester Comprehensive Cancer Center, Miami. Please welcome.
Well, thank you for the kind introduction, and I've enjoyed working with you guys and working with the entire SPAGN group and all of the advocates. It's really a critical interaction. As a scientist, a clinician, a clinical researcher, we have to get our direction from the advocates. That's how, and the patients. That's how we understand what's important. So this is a very important meeting, and it's been a delight to see it continue to grow. So I was given several topics to discuss, and so.
You have 60 minutes.
So yeah, somehow an hour. I don't know if I can talk that long, but we'll have some good discussion for sure. And I'll go over some of the areas that I think are important and some of the areas where advocacy, patients, clinicians, scientists all really interact together. And so I do clinical research with a number of different entities and with our institution. And I'll start by discussing a little bit about immunooncology and immunotherapy and immunology in general. And I hear all the time from clinicians, sometimes sarcoma clinicians, some of my colleagues, sometimes community oncologists, sometimes experts in kidney cancer, that immunotherapy, immunooncology, immunology doesn't have a role in sarcoma. And I hope I can convince you today that we've actually made a lot of progress. We certainly have a lot to do, but that we're making progress in this area. And I think it's quite likely it's the tip of the iceberg. We've been using chemotherapy in cancer for 70 years plus, some of the same chemotherapy we use today. We're just now using immunology, immunotherapy. It's all very new. We're learning how to use it more efficiently and better, and things I'm convinced will continue to improve and get better. So let's talk a little bit about the immunology, just to get some nomenclature and some vocabulary. On the left side, the type of immunity is innate, meaning it just happens. It's just things that happen reflexively. If you get an infection, there can be release of complement cytokines, chemokines. There's no regulation of it, there's no change of it. They just get released and then they go away. They do their thing and go away. Those are chemicals. And then just to the right are the cells, the cellular compartment of what's called innate immunity. And this is a primitive immune system that doesn't really react, it doesn't adapt. When you get a bacterial infection, neutrophils proliferate and they fight it and they release chemicals. But they don't learn anything, they don't shift, they don't adapt. So that's called innate. Macrophages, they eat up the material that from the bacteria that the neutrophils killed, and then they present it to some of the cells on the right through the dendritic cells. And then these other ones are involved in allergic reactions, parasitic infections. But the innate means that it doesn't really adapt or change over time. Now on the right, we have the adaptive immunity. Adaptive immunity, by the name, changes over time. It can adapt to a changing tumor. It can adapt to a specific antigen on a cancer cell and attack that one with specificity. And it's coordinated with the innate cells, such as the antigen-presenting cells. When there's an infection or even a cancer in the body, the neutrophils and macrophage and dendritic cells process it and present a protein out on their surface called an antigen. So when we talk about antigen, that's a metabolized protein from, in this case, what we're talking about today, a cancer cell. A cancer antigen is a protein from a cell. And the antigen cells present that protein and show it to the T cells and the B cells. So we have a vast number of T cells and B cells, and they're all different. And we have millions of different T cells and B cells in our body. Each one is different. They recognize different antigens. So when this antigen-presenting cell shows the protein out there for the T cells and the B cells, there may be one T cell that recognizes it, maybe two, oftentimes one out of those millions in your body, only one recognizes that antigen. But when it does, in a normal immune system, a lot of things happen really quickly, a lot of changes, and it's the adaptive changes to that specific antigen being matched with that specific T cell or B cell. Well, when the T cell is activated by an antigen, it does different things. There's a subset of T cells called CD4, and they regulate the immune system so that you don't get, for instance, an autoimmune disease. They calm it down, they control it. The CD8 cells are the cytotoxic killer. They used to be called killer T cells. So they kill whatever has that antigen. So in theory, if this antigen presenting cell shows a tumor antigen to the T cell, if the T cell gets activated, and then the T cell searches your whole, and then it proliferates. You go from one T cell to millions of T cells that all recognize that same antigen. And they scour your body trying to find that antigen. If it's a cancer cell, it's a protein on the cancer cell, and they kill it. And so the B cells, on the other hand, they don't actively kill tumor cells or bacteria or viruses themselves. When they get shown an antigen and bind to it, they also get active, and one B cell might go to millions. But those millions of B cells don't directly attack the tumor, they produce antibodies. And these antibodies can then attack the cancer cell or the bacteria or the virus or whatever is carrying that antigen throughout the entire body. So you can so those are the players roughly that are in immunology, immunooncology, and of course immunotherapy. And you can visualize it like this. So we have a tumor cell at the bottom, and the tumor cell has on it some antigens. In sarcoma, there are a lot of antigens that are unique in the human body. Just have KIT mutations. Normal cells don't have KIT mutations. Chondrosarcomas, some of them have IDH mutation. Normal cells don't have a mutant IDH protein. Ewing sarcoma has EWS Fly1 translocation. Desmoid tumor has beta-catenin mutation. And so these mutations, translocations are unique. They're not on normal cells. So they're called neoantigens or new antigens, and they're on the tumor cells. To me, that gives us a lot of hope that we can figure out how to get the immune system to attack these tumor cells that have proteins on them that aren't on the normal cells. So it can provide specificity because we don't want to treat the immune system in such a way that it attacks normal cells. That's autoimmune disease, diabetes, it could be any other multiple sclerosis, other autoimmune diseases. So the tumor cell, if you look at the bottom of it, it says AG, that's antigen, and it's showing it to the T cell. Like, remember we were talking about the T cell can see that antigen and bind to it. But for the T cell to get activated against that antigen to attack it and to kill the tumor cell, there has to be these co-expressed proteins, these co-receptors that have to get activated too. And if you look in the middle to the right of the T cell, you'll see some things like CTLA4, PD1, and then on the tumor cell, PDL1. These are co-proteins that are co-activators. They're called co-activators. And to avoid autoimmune scenarios where your immune system's attacking your body, the T cell's set up that you have to have both. You have to have an antigen, and you have to have activation of a co-activator. It's like, it's like the nuclear bomb. You want to have two switches. You have to have two separate switches to activate it because you don't want it to go crazy, okay? You don't want an accident to happen. So these both have to happen. When it does, the T cell proliferates. It splits from 1 to 2, 2 to 4, 8, 16 to millions of T cells. The cytotoxic T cells then can kill the bacteria, the virus, the tumor cell, the yeast, whatever its antigen is recognizing, it can kill. Now I mentioned these co-activators, PD1, CTLA4, and PDL1 is on the tumor cell. I mention those because tumors can evade our immune system. They wear a camouflage, and our immune cells can't see them. That camouflage consists of inactivation or loss of the co-activators, so that the antigen is there and one switch gets flipped, but the second switch cannot be flipped because the second co-activator isn't recognized by the T cell. It can't find the PDL1. The CTLA4 can't bind to the CD80. It's blocked or it's downregulated by the tumor cell. Tumor cells, they're not really smart, but they're evolutionary. So tumor cells, we may be getting tumor cells in our body all the time, but our immune system kills them. When the immune system doesn't, that's the one that survives. It's evolutionary. It's selection. And so that biology, I mean, it took like 60 years to understand that biology. Jim Allison got a Nobel Prize for it. And then once the biology was understood, pharmaceutical companies started developing ways to activate these co-activators. So when you hear people talking about immunotherapy, we're usually talking about immune checkpoint inhibitors. It's called a checkpoint because if it doesn't happen, the immune system doesn't go forward. It doesn't get activated. It's stuck there. But we have these antibodies now against CTLA4, against PD1, against PDL1 that can activate that checkpoint by stimulating the co-activators and in essence removing the camouflage from the tumor cell. So now our immune system can see the tumor cell and can attack it and recognize it as being foreign, not being part of you, but being of the patient, but being foreign. And so these are the drugs that we're talking about, like pembrolizumab, ipilimumab, nivolumab, atezolizumab for ASPS that we're using and is FDA approved for sarcoma, for ASPS anyway. And so these are the agents that are available that are showing some early activity in sarcoma subsets, so select types of sarcoma. And this is the mechanism of how that's happening. Now you'll notice there's a lot going on there other than the PD1, other than the PDL1. There's the dendritic cell also. We're not even talking about B cells and vaccines. So there's a lot of research in these areas that give us a lot of hope for the future. And this is really a cross-section of where we are today. There's the research that's being built on this with combination therapies, with different approaches. We recently had a publication accepted that was combining a PD-L1 with a CTLA-4 together, both of them, in patients with soft tissue sarcoma, and showed about 25% of the patients had substantial decrease in their tumor size. And so there is certainly some hope for the future with immunotherapy. I'll show you some evidence of how we're using it today. So first time I got involved with immunotherapy was actually about 15 years ago in sarcoma. And with one of my colleagues, Lei Chen, designed a clinical trial using interferon alpha. Interferon alpha is one of those chemicals that's in the immune system that activates the CD8 killer cells. So it turns on the killer cells. So patients received that plus imatinib for their GIST, and all 8 patients responded to their imatinib in the setting. In the biopsies, there were increased CD8 cells in the tumor samples. Since then, Arun Singh did an Ipi-Nivo study. This is a CTLA-4 co-activator plus a PD-L1 activator in GIST, and showed 2 patients responded clinically. Suzanne George looked at leiomyosarcoma. Sandra D'Angelo looked at imatinib ipilimumab and showed some responses, not in size, but some internal characteristic of the patients. And Melissa Burgess with pembrolizumab published SARC-28. This is one of the SARC trials. SARC is our clinical trial arm of the sarcoma research community. And this was really the first large trial in patients with sarcoma looking at immune checkpoint inhibitors. This one is pembrolizumab. It's a PD-L1 activator, so it activates the immune system. And so this study showed some activity in what's called, some people call UPS, which is an undifferentiated pleomorphic sarcoma or a poorly differentiated sarcoma. Some institutions will call it fibrosarcoma, myxofibrosarcoma. Years and years ago, it used to be called malignant fibrous histiocytoma. All the same entity, but in that study, over 20% of patients had decrease in their tumor size. So the patients that have these pleomorphic sarcomas do seem to have some benefit from checkpoint inhibitors. A lower benefit was seen in the patients with dedifferentiated liposarcoma. Seth Pollack did a phase 1 study combining doxorubicin and pembrolizumab in sarcoma patients, and it looked very promising. So he has been able to open that nationally in the US in an alliance trial that is phase 3 comparing doxorubicin plus pembrolizumab to doxorubicin alone as a single agent for patients with the UPS, myxofibrosarcoma, pleomorphic sarcoma entity. He's considering adding and expanding to dedifferentiated liposarcoma. That trial is ongoing throughout the US. If anybody knows of patients with those types of sarcoma that are looking for therapy, Breland Wilkie, who's in University of Colorado, did a phase 2 study combining axitinib, which is a kinase inhibitor, not an immunotherapy, with pembrolizumab, again, this PD-L1. And so axitinib changes the tumor microenvironment. So it binds to blood vessels in the tumor, makes the tumor become hypoxic, changes the chemicals that are in the microenvironment that are suppressing the immune system. And so when patients take axitinib, the theory and the current theory is that it increases the ability of the immunotherapy to get into the tumor, to get to the tumor cells, and modifies the microenvironment so that the immunotherapy is more effective. And in fact, in this study, 66% of patients had stable disease or better for 3 months, and the subset of patients with alveolar soft part sarcoma had a 50% response rate. And so then some of the patients on that study, a couple of them had angiosarcoma, and we saw really good responses. So we looked back retrospectively on our series of angiosarcoma patients treated with checkpoint inhibitors and found that even though these patients treated had already had all standard therapies, they had all standard sarcoma chemotherapies, targeted therapies. They came to us because they had no other therapeutic options. We treated the angiosarcoma patients with checkpoint inhibitor, and 71% of the patients had their tumors decrease in size. Maybe the highest response rate in all of solid tumors to immunotherapy. So immunotherapy can be effective for sarcoma patients. It's just a matter of, with the tools we have today, it's a matter of finding the right patients. Hopefully, the tools we'll develop tomorrow will increase this benefit. One of those tools for therapy is called antibody-drug conjugate. And this technology takes an antibody against a protein on a tumor cell. Let's say a tumor cell has a novel protein X on the surface. In the laboratory, scientists can make antibodies against protein X that bind to it. And if X isn't present on normal cells, it's specific then for tumor cells. Antibodies, though, alone don't often kill tumor cells or normal cells. So what has happened over the years is technology to stick toxic agents on the antibodies. And this is called a payload because the toxin, in this case topoisomerase 1 inhibitor, has a linker that releases it when it gets into the cell. So the antibody can bind to an antigen or a protein on the tumor cell, and then it can be taken into the tumor cell for normal processing. Tumors do this all the time, called endocytosis. But then once it's in that acidic environment, the toxin gets released from the linker and is able to get into the DNA, bind to the DNA, and kill the tumor cell. And this provides specificity. It also is already making a presence in sarcoma. There are 2 clinical trials that are nearly open at our institution and at several other sarcoma centers around the country. In the trials, one binds a surface protein called UPARAP, and another one a surface protein called KIT for GI stromal tumor patients. And so these are exciting. We have very little experience with these agents in sarcoma, so we're hoping to provide specificity, safety, and increased benefit to cancer patients. Let's talk a little bit about cellular therapy. Cellular therapy, if you remember the diagram, it's taking the T cells through a process called leukapheresis. T cells are taken out, blood is taken out of a patient. T cells are isolated using these coated beads. Then the T cells are activated by these interferon, these other chemicals I talked to you about that can turn a T cell into a cytotoxic CD8 killer T cell. This process of activation takes cytokines such as IL-2, interferon, to make the T cells grow from 1 to 2 to 4 to 8 to 16 so that there are millions of T cells and that they're activated and they have that killer T cell phenotype. And then once they're activated and expanded, then they can be infused back into the patient. So it's the patient's own T cells that have been activated. But what has to happen first is the patient has to have some chemotherapy to decrease their existing T cells and then infused with the activated T cells that are activated against their tumor. And the T cells can then further expand and fill up that space in the blood that was evacuated by the T cells that were decreased by chemotherapy. So that there are some examples like synovial sarcoma where this technology has been used in a clinical trial and led to FDA approval. So today, we have access to this drug called afami-cel, and afami-cel is approved for synovial sarcoma patients who have a neoantigen called MAGE4. So they have to have this protein on their surface because that's what the T cell recognizes. But then they also have to have a specific protein on their normal cell, on the tumor cell, called HLA-A2. And maybe 50% of the population is HLA-A2. If you don't have HLA-A2, then this approach with this cellular therapy is not effective. We need better therapies that are not HLA restricted. So this is the activity of afami-cel in synovial sarcoma. And this diagram, this chart on the left, it's called a waterfall plot. And each bar that's going down represents a decrease in tumor size. The numbers out on the left are percentages on the y-axis. And so you can see on the far right of that chart, the two blue bars that go all the way down to the bottom had 100% decrease in tumor size. That means these patients had a complete response to therapy. Tumor was gone completely, which is rare in sarcoma. It's rare in a lot of solid tumors. But you can also see that the majority of patients had some shrinkage of their tumor. So this is one of the first, it may be the first cellular therapy that's approved for solid tumors. And it's the first therapy approved specifically for synovial sarcoma. Lymphoma people have been using these medicines for several years in the hematologic space, but this is a solid tumor. And in fact, 43% of the patients had what we call a clinical trial response. A clinical trial response means the tumor shrank by 30% or more. I would argue that if a tumor shrank by 29%, that should be a response too. That's a good thing, right? But you can see on the waterfall plot that the majority of patients did have some decrease in tumor size. Now there's another type of trial that we have open at Sylvester that it's not a T cell, it's a different cell. It's the innate side. It's called NK cell, natural killer cell. And it's a more primitive, non-adaptive cell, but they can be trained in the lab to attack the tumor cells. And this trial is an NK cell trial called TINK that is available for sarcoma patients. So in conclusion, immunotherapeutic approaches do benefit sarcoma patients. With the tools we have today and the approaches, angiosarcoma, ASPS, UPS, D-diff lipo, these patients, I have no reservation treating these patients with immunotherapy approach. We know that there is some potential benefit, sometimes long-term, especially in angiosarcoma. Some patients have been cured. T-cell therapy is effective. We have FDA approval for synovial sarcoma. That should not be overlooked. We have to work with advocates. We need to work with advocates because community oncologists don't always know this. A patient with synovial sarcoma might call or text or email SFA and start a dialogue and realize that their community oncologist never even talked to them or knew about afami-cel. And so from the most recent studies, about half of sarcoma patients are treated in the community. Only about half go to a major sarcoma center. And so advocates, patients, and advocacy groups can help spread the word about these treatments, because this is a lot of information I've gone over with you today, and a busy community oncologist that might see 1 or 2 sarcoma patients a year can't keep up with this literature. So the patients may reach out to you asking for advice, so it's important. It's an important role. So As I mentioned, there are a lot of other neoantigens other than MAGE-4. There's a lot of other ones out there that are specific to tumor cells, tumor-specific antigens in sarcoma. And so there's going to be additional progress, I'm certain. We're still in the new phase of immunotherapy. We're using single agents. We're starting to use some combinations of two. It may take more. It may take 3 agents. It may take tyrosine kinase inhibitor, immunotherapy, chemotherapy, and different agents at different times to get the optimal response to therapy. So I'll talk a little more about combinations because we are entering that era. And I'll share with you some of the recent data on combination therapy. We've used this for chemotherapy in sarcoma. Combination therapy is effective. These are some of the older chemotherapies, doxorubicin and ifosfamide. As single agents, 20%, 35% response rates, but when you combine them together, 65% of patients get a decrease in tumor size. And again, a substantial decrease by 30% or more. And so combination therapy has been a paradigm in sarcoma patient management for many, many years. These publications go back to even the 70s and 90s for some of them. And so let's talk about one of the first combinations that emerged, and that was combining tyrosine kinase inhibitors with other therapies. The first tyrosine kinase inhibitor activity seen in sarcoma was in alveolar soft part sarcoma in using an agent that's not approved but was used in a clinical trial called cediranib. It's a multi-targeted tyrosine kinase inhibitor. It targets some proteins on the tumor cell, but it also targets some proteins on the blood vessel inside the tumor. And so by targeting the tumor and the tumor's blood supply, it's thought that these agents have some activity in alveolar soft part sarcoma. And if you look at the response rates, they're around 25-29% of patients get some benefit with ASPS from a tyrosine kinase inhibitor. Alice Chen and her group and others at NCI looked at immune checkpoint inhibitors in alveolar soft part sarcoma. We discussed some of this data already. The response rate was about 37%. So that made us think that perhaps combining the two could provide even more benefit. And we're always trying to find a way to cure patients. So this is the first combination study looking at a tyrosine kinase inhibitor plus a checkpoint inhibitor in sarcoma patients. And this study in ASPS showed a 60% response rate. About another 30% of patients had some shrinkage but didn't meet that 30% criteria. So when I see a patient with alveolar soft part sarcoma, like I did last week, who really had a large tumor, painful, we needed to get it shrink. It's a primary tumor. If we can get it to shrink, there might be a surgical cure, possibly with radiation therapy. If we want to get the tumor to shrink, give the patient the best response, we would use the combination of axitinib plus pembrolizumab. If a response is, for whatever reason, is not the goal, then a single agent, such as enlotinib, pazopanib, axitinib, or single agent immunotherapy, pembrolizumab, is reasonable, but we generally prefer the combination at our center. A second study validated this with 2 different agents, sunitinib plus nivolumab from the Spanish sarcoma group, Javier Martin Broto, in the ASPS patients. Other sarcoma patients were on here and also seemed to have some benefit as well. So immune checkpoint inhibitor plus a tyrosine kinase inhibitor is clearly more active than single agents. We don't know whether that helps patients live longer or not, but if the goal is to shrink the tumor, the combination is the way to go. We're very excited about antibody drug conjugates and the specificity and activity and promise in sarcomas, as well as additional cellular therapies, and hopefully ones that aren't as restrictive with HLA and the requirement of the protein in the future. So let's talk a little bit about innovations and some of the direction we need to go with clinical trials. And this is me in some ways criticizing my community, so don't tell anybody outside this room. But we've not designed the best trials, in my opinion. Some of the trials I'm going to discuss, I didn't participate in, our center didn't participate in because we didn't like the design. And we need to design better trials because some of our trials may have 400 or 500 sarcoma patients on the trial. We have an ethical obligation to provide the best trial possible, and I believe we have some room to improve here. And this is a table summarizes clinical trials in the sarcoma space that have new agents, brand new treatments that have in the front line that have been compared to single agent doxorubicin alone. Remember, at our center and many across the world, combination therapy is more effective. And so we are not necessarily always supportive of single agent doxorubicin studies. These studies had that design, and this represents 8 studies. Okay, this is a lot of patients. This may be 1,000 or more patients. All of these studies were negative. The experimental therapy was not superior to the doxorubicin alone. And you have to wonder, maybe we need to be a little smarter about how we design our trials. It's challenging for us in sarcoma because it's a rare disease. How do I design a trial with enough patients when the type of cancer is so rare? Well, we have to do national, international trials. Those are much more expensive. How do we get pharmaceutical companies excited about rare diseases? It's not always easy. But we need to step back and think about how to design better trials. We don't want to keep trying out new agents and coming up empty-handed. We want to be able to get FDA-approved agents, EMA-approved agents, so that patients can get access. So rather than first-line studies compared to doxorubicin, my opinion is that we should be doing later-line studies. Later-line studies generally are more successful in sarcoma. These are 4 studies, late-line studies, that were compared against other agents, such as dacarbazine, in one case a placebo, which is different conversation, but comparing to dacarbazine, comparing to gemcitabine. These were all positive studies. Some of them led to FDA approval. They're all listed in our NCCN guidelines. And they got there because the study design was a lot better. And so taking these agents like aldoxorubicin, like brigamadlin, why try to put it in front line against something that we already know is effective? Let's use it in later line therapies where we have patients that need new treatments. So some of the directions and opportunities we have are to look at single histology studies, to look at the novel agents in later line. Like I mentioned, trabectedin and eribulin, we got those FDA approved because they were used in later line therapies where there's much fewer and less active treatments, but it's where the biggest need is. And also, we should not be doing first line comparator to doxorubicin alone. Doxorubicin plus ifosfamide is more effective. It's the standard of care at most sarcoma centers. Single agent doxorubicin also introduces a selection bias. Lower grade tumors, tumor patients with smaller volume tumors, and it's really shouldn't be using an endpoint of progression-free survival with single agent doxorubicin. The other thing that's imperative is biomarker-driven studies. There's been several studies in the last 5-6 years that were negative because a biomarker that was known was not used. It was not used. Patients were unselected and treated with a therapy we knew wasn't going to be effective. So biomarker-driven therapy is really critical. We have brilliant sarcoma scientists around the country. We can make more progress here. And the last item says we should maybe get away from single agent doxorubicin, and we sure shouldn't be using placebo. We're in the area of artificial intelligence, and we should be developing synthetic placebos. We've been taking care of sarcoma patients at our centers for 30, 40, 50 years. We know what's going to happen with single agent doxorubicin. We have those records. Let's design a synthetic control arm so that a patient with a malignant cancer doesn't get randomized to a placebo. Speaking of placebos, we did this project with Cesar Serrano, he led it, and we looked at just studies, GI stromal tumor studies, in patients that were treated on placebo trials. And so these patients were these 5 studies, patients were randomized to either a treatment or a placebo. And if you look at the graph, the blue curves are the patients, represent the patients that were treated. Now on this type of diagram, as you go from left to right, every downward deflection represents the time it took for a patient's tumor to start growing. And so the orange and yellow or gold curves, lines, you can see that time is a lot shorter. It's at like 2 months, 1.5 months, 2 months. The patient's tumors are starting to grow a lot faster. Than the blue curve, it takes longer for patients' tumors to grow. And that's represented in the table just to the right of the curve, or above to the right of the curve, where you can see the studies listed and what's called the median progression-free survival, or approximately the average amount of time it takes for half of the patient's tumors to start growing. And it's much longer. It's much longer. For instance, the first one, sunitinib, it's 4 times longer. And so the reason we did this is because we don't think we should be giving patients placebos, period, is the reason that Cesar and I advocated for this analysis. And the pharmaceutical companies were willing to work with us on this data to some degree. What's even more disturbing is that this is overall survival. So this is how long patients lived. And so these patients were put on placebo studies, and we know that if they were given the placebo, they didn't live as long as a patient that got the treatment. And I didn't participate in any of these studies because I don't believe ethically in placebo studies for patients with malignant cancer. And so this really proves the point and underscores the concept that we need more effective agents, but we shouldn't be doing placebo studies in today's day and age. It's not just the efficacy, it's also the side effects. These are side effects that patients got when they were getting placebo. When we give patients a treatment for their cancer, sure, there's going to be some side effects from the medicine, but we expect, if they respond, we expect the cancer-related side effects to become less. That's the reason, one of the reasons we treat patients with cancer is to decrease their symptoms. Well, these patients had symptoms related to their cancer on a placebo and were not getting those symptoms treated. Another reason for us to move on from placebos. So in the future, we're hoping that placebo would be replaced by some type of artificial intelligence, machine learning derived approach, so that a synthetic control arm can be used instead of a placebo, especially in patients with malignant sarcomas. So let's talk a little bit more about artificial intelligence and machine learning. So these are being used in medicine. Our sarcoma pathologists are using them already to help them analyze patients. Our radiologists are using them. And this really allows pathologists and radiologists to be able to review more tissue samples, help more patients, because With rare diseases, our radiologists and pathologists need that type of support. It also, radiology and pathology provide vast amounts of data. Just like genomics, there's radiomics and pathologyomics, where that data can be taken out in discrete pieces and analyzed in the bigger picture. And Antoine Italiano's group published a review article on this. So here's a project that we did looking at a drug. So we looked at drug sensitivity and we looked at inhibitor activity. So we took the cell lines, put them in the lab, exposed them to different drugs, several hundred different drugs, developed a drug sensitivity score, and then used in silico theoretical approaches with kinase. What is the probability of kinase is going to inhibit the target? Is the target present on the tumor cell? And then merge that data to identify tumor cell lines that have the target and have the drug sensitivity. And this much data, this much raw data can only really be processed through artificial intelligence and machine learning to identify new drugs. Here's another way we looked at it in PET scan. Russ Cooker looked at using AI and machine learning to improve PET CT activity. And what's done is individual pixels on the PET scan are abstracted before and after treatment. And this was 165 different parameters for one PET scan. And in this study, he found that there was 99% congruence with the response to treatment when it was done by artificial intelligence compared to living, breathing radiologists. And so this needs to be certainly validated prospectively, but it's compelling that these types of approaches will help our radiologists be more effective and be able to help more patients. And taking better care of the patients is the ultimate outcome. Let's talk a little bit about leveraging technology. One of our surgeons, Fran Hornacek, works closely with a surgeon who also leads a bioengineering department in Ecuador. And so this is a patient from Ecuador on the far left that has a pelvic sarcoma. In this case, the sarcoma you can see is in white. You see a lot of gray, you'll see a lot of black. What's bright white in the middle is the sarcoma in the pelvis. And so that information can be input into a processing software platform that can recreate it in 3 dimensions on the computer screen and give different tissues different colors. So the pelvic bone is orange, the sacrum is green, and the tumor is purple in this recreation from the MRI. So from the MRI to a 3D creation, and then goes to another platform that can do the 3D surgical planning. So up in the top right, you can see the pelvis, you can see a purple tumor. The purple tumor, you can see the area where the pelvis would be removed, which is in red right next to it. So that red area is the area the surgeon needs to remove to be able to remove the sarcoma in its entirety without spilling anything, without cutting into the tumor, a curative approach. And then at the bottom right is the reconstruction guidelines. So these are the cutting guidelines in the area to be reconstructed. And then all that data is sent to a 3D printer, and the 3D printer prints out a replica of the patient's pelvis so that at the bottom middle, so that everyone, including the patient, can, if they want, can see what's going to happen at the time of their surgery. This is what's going to be removed. And then the 3D printed prosthesis will be put back in its place. And this, in this situation, we're doing it for educational purposes and to create the bioprosthesis. But this is an example of collaboration across institutions and even international collaboration, which we have to do in sarcoma. Spain is a great example of that. We have to have international collaborations to advance the field, especially in our rare types of cancers. So international is key. I'll say a little bit about genomics and therapeutic decision-making. Just like we're getting all of those radiomics and information from pathology, we have vast amounts of information from genetics from patients that are having their tumors tested. One example is GI stromal tumor, and these are the different molecular subtypes. Each molecular subtype is treated with a different approach. So I suspect one day, hopefully soon, we'll have leiomyosarcoma subtypes, molecular subtypes that are treated effectively in different ways. Same for osteosarcoma. We hope to be able to continue research. It may not be genomics like this, it may be epigenetics. There may be epigenetic signatures. There may be other factors that help us molecularly subcategorize sarcomas to lead to better therapy. Unfortunately, in the GIST world, and I suspect in other sarcoma types, mutation testing is not being performed enough. And so there is a type of translocation, a type of genetic event that involves a gene called TREC. And it can affect a lot of different types of cancer. The number one solid tumor is sarcoma. So one could argue that maybe every single sarcoma patient should undergo genetic testing, especially if they need treatment. And we call this next generation sequencing or NGS. But in the US anyway, it's not. Only about 30% of GIST patients are being tested. And it's probably less for other sarcoma types. And it's important because if you identify a TREC, or if you, in this case, you identify a RAF mutation, you can use a RAF inhibitor. This is an example of TREC. If you identify a TREC mutation, translocation, you can use a TREC inhibitor, larotrectinib. And these TREC translocations occur across a broad array of cancer types, including several different types of sarcoma. So these really, you don't know if somebody's tumor may be driven by this unless you look for it. The only way to look for it is mutation testing. So NGS, or mutation testing, is really required to optimize therapy. Other types of newer testing beyond testing the tumor is testing the mutations in the blood, called ctDNA. We don't really even have time to talk about sarcoma cellular metabolism. This is an area that is more recently being researched, and some treatments are coming. But we've been talking a lot about mutations and translocations. That's a lot simpler than metabolism. Metabolism is very complicated. There's multiple pathways, there's redundancy, and there are some sarcomas that are driven by oncometabolites, such as IDH mutated chondrosarcoma and SDH deficient GI stromal tumor. And understanding these pathways is the only way really to lead to more effective treatment. I mentioned global collaboration because Spain is a great example of that, and it's critical. And these are some of our multinational progress that we've made. On the left, our professional societies and others, and foundations. And importantly, increasing global access to medicines is really critical, and it's not being addressed. And there are areas in the world where there is no access. Patients have sarcomas and don't have access to drugs. We need better quality generics, biosimilars. We need to have affordable pricing, especially in areas where it's not really affordable. We, as clinicians and everyone here as advocates, have to continue to put pressure on the healthcare government the funding agencies, whether it's universal coverage or other approaches or the ATOM coalition. These are all areas where we need to make progress for the global care of sarcoma patients. So in summary, research-driven new combination therapies are paving the way for improved outcomes. And I do emphasize combination therapies. There are going to be sarcomas that are not cured with one approach. Technology is accelerating, bioengineering, artificial intelligence. We need to, in sarcoma, we should be leveraging those technologies as much as we can and becoming early adopters, not waiting. Continue to expand our global collaboration. And I honestly believe with all of this progress that's ongoing, the future is bright and getting better for sarcoma patients. So I'd like to thank everybody for attending this very long session, and thank you for listening to me talk a lot, and I would be happy to answer any questions. Thank you.
Thank you, Dr. Trent. That's a lot to unpack. It was great information, fantastic. Are there any questions from the audience for Dr. Trent?
Oh, hi.
I wanted to go back to when you were talking about the immune checkpoint inhibitors. All the successes you have from most of the sarcomas that are immunologically high, clear cells are cold, and the physicians keep prescribing these checkpoint inhibitors over and over again unsuccessfully.
Right.
And I know there's plenty of push to try to come on top of some of these other ideas, and we've actually seen like radiation binding that kind of almost accelerates it sometimes. I don't know if you can talk to any success that you've heard about turning any of these tumors larger or
Yeah,
other ideas.
Yeah, clear cell sarcoma is a tough one. It's a tough one. It's a challenge. We were very optimistic about checkpoint inhibitors since it used to be called melanoma, malignant melanoma of soft parts, but it's really not a melanoma, it's a sarcoma. And it is a cold one, so, and there have not been great treatments for it, but it doesn't mean there won't be. Alveolar soft part sarcoma is also considered a cold tumor immunologically, but you end up with like a 35% response rate to checkpoint inhibitors.
I think it seems like there's maybe there's just a percentage of them that are
Yeah. Yeah, so so GIST is also a cold tumor. And so in this older study we did, we used interferon. And so maybe there's actually some trials going on combining checkpoint inhibitors with other agents to try to make the tumors hot. And so we need either some trials for clear cell or some sarcoma trials that make sure to get clear cell on these studies, or solid tumor trials. So at our center, we have a lot of solid tumor trials that are for any solid tumor, and we try to get clear cell sarcoma patients on those studies to try to get something to help checkpoint inhibitor or other therapies. But you're exactly 100% right. I think combinations is the only way we're going to be able to do that.
we agree. We've got a phase 2 trying to get started.
Well, I'd like to hear more about it.
Yeah.
Yeah. Yeah.
Thank you very much for the so my name is Steve from me, and I'm means so how the access especially for access to sarcoma cases for the early diagnosis. So in India, people like same concerns from civils, especially in the remote locations and rural areas. So that supports a lot for the early diagnosis. If I can share about myself, so I got those posts are in, and right in the school got replaced with this type of capability. And a lot of like thousands of cases like was not available. So maybe in the other places as well, telemedicine can.
Yeah. Thank you.
So first of all, thank you for a fascinating talk. How do you see the medical community accepting results or recommendations from AI systems, especially with respect to
That's a good point. The way we're leveraging it is to use it to make people more efficient. Help the radiologists become more efficient and the pathologists, but not replacing them by any means. We're nowhere near, and I don't know when we will be able to replace a pathologist with artificial intelligence. It's more of maybe increasing efficiency.
And you see the medical community accepting this?
Yeah, I think so. Cautiously.
Your group is advanced in that respect?
I think very
It's a general trend you see.
Yeah, we don't want, I don't think any of us would accept an AI-only diagnosis. We would want a sarcoma pathologist to have reviewed it and agreed with it. And so, but I think that that's what's going to happen in the future, whether we like it or not. Yeah.
So I have two questions. One is immunotherapy. Have you gotten a handle on, what works and what doesn't work? What subtypes immunotherapy would not be effective? And the second question is, I was wondering if you could elaborate more about non-placebo studies and what they would look like, and how you can convince regulators and everybody else about not using placebos.
That's where the rubber hits the pavement, right? You have to have a regulatory agency agree to it.
Right.
And they've said that they're open to it. We have to, and it's not just the regulatory agency, it's also the pharmaceutical industry has to agree to it. Even if we agree to it, both the pharmaceutical industry and the FDA have to both agree to it also. But they have said publicly that they're open to it. They're open to non-placebo trials, active comparators, synthetic controls, but I have yet to see one happen. So I don't know what direction that'll go.
So the question specifically is, what would you use to substitute placebo?
You would use it's basically similar to a historical control. So AI can analyze, millions, billions of time points and pieces of data and provide a more accurate historical control. What we do in phase 2 trials is we say, well, every time we've treated, we don't have any active drugs against clear cell sarcoma. So in our phase 2 trial, we're going to say if we get a 10% response rate, we're going to call it success. And it's going to be a successful study. And it's going to be positive. And that's based on statistical design saying that we don't think any drug is ever going to have a 5% or higher. Historically, there's never been a 5% or higher response rate with a clear cell sarcoma when you look at maybe 100 of them. So the synthetic control would be using more data, having more certainty in that comparator number, because we're always comparing it to something. the two response rates or the two PFS. It's always being compared. And so instead of a placebo, where we know the response rate's going to be 0, we would have a synthetic control that statistically creates a number, let's say 3% or 4%, based on a lot of data, based on modeling, repeating modeling so that it learns, and designing a number that everybody would agree on. And so that's what it would look like.
Go ahead.
In the same line of what you were asking, can you talk to the use, implementation, and acceptance of other models, such as the organoids and spheroids and in vitro cell-based assays that ideally could replace also animal models? How far along are those, and what are the odds? How are they being used, and what are the odds? These are going to be eventually replacing the current animal models treatment.
I don't know the answer to that. I'm not a big fan of animal models. I would like to see organoids replace it, but, and also most of the animal models, the tumors don't have an immune system. So studying immunotherapy in an animal model is very, very challenging. There's some that are being created, reconstituting animals with human immune system, but it's never gonna be 100%. So there's always gonna be limitations to animal models. But, what concerns me more is maybe that there are animal models that don't show benefit when in a human they would, and we're missing out on good drugs. And so if there were spectacular results with an organoid or with some other model, I personally would think we would try to make the case to move it into clinical trial. There are some sarcoma types that don't have animal models. So for instance, dermatofibrosarcoma, we just started treating patients with imatinib because it made sense. And then published a series, and now it's on our NCCN guidelines. But there was never any animal model or organoid or anything. So it is possible. It's just it's just the paradigm that they like to see. Like for instance, our ETCTN, one of our national early phase networks, they almost always require 2 animal models for the government to fund a study of a certain drug. So it's a challenge. It really is. And I share with you, I don't think we always need that many when we have patients that need treatment. So I don't have a good answer.
I work with the startups on that early stage side, and it's difficult to know if they're worthy of being funded if downstream no one's going to listen to their results, they're fine. So if people still demand and expect animal studies, all the companies will pile up and get stuck right at that point.
Yeah.
It's guy in the water.
Yep.
Well, the good news is you're in a room with patient advocates. So when we find a better way, you just activate us. I have a quick, 2 questions. One is you talked about trial designs. And you showed the slide of all the trials that failed, which we know a high proportion of trials fail. But one thing that we didn't get into, and I'd like to hear your thoughts on this if you can speak about it, is the trial designs that are forced upon us by regulatory, and I think it's good for it. And what I mean by that is the endpoints sometimes, right? And not flexibility in terms of endpoints. And when a drug actually shows activity, but it doesn't meet the p-value, so therefore it's considered a failed trial. I mean, we could go on and on about them. But as we think about advocacy and we think about how we can affect, we've had so many conversations during this meeting about clinical trial designs and where patients can partner and be involved. And this is one of these areas where we can potentially be involved going to regulatory and saying some of these things have to change. Anyway, can you speak a little bit about some of the challenges we face just because of that?
Yeah. The p-value drives a lot of us crazy. It really does. You can have, especially in rare diseases, because it's hard for us to do a trial of 1,000 patients. You do the most patients you can do, and you see a separation. You see that drug X is 20% better than let's say drug X is 19% better than drug Y. But when you design the study, you were expecting it to be 20% better. So your p-value is 0.07. And then everybody calls it negative, even though it's really not, you know, it's a challenge. So the statistical designs, and we should design better studies so there's less likelihood of that happening. All those doxorubicin studies are not surprising. When you go back and look at it, it's not surprising. Compare aldoxorubicin to dacarbazine. Those patients need a new drug, and if aldoxorubicin helps them and it gets FDA approved, that's a good thing. But why on earth try it against something that's already good? And so our design of the study, we can design studies that are less likely to result in that. And then alternative designs like continuous reassessment methods, CRM designs, and Bayesian designs, you can keep adding patients until you hit that statistical significance. So Bayesian designs are a smarter way also to do it.
Yeah, which leads me to our next question, because one would argue, and I'm going to use Brigham-Madeline as an example of a drug that showed activity, didn't meet p-value, you know, but it, you know, when you look at something and then some of the doxorubicin, you know, doxorubicin it works, it performs, but giving the patient the choice, right? Because of the long-term effects of chemotherapy and doxorubicin, you know, from a patient advocacy perspective, there may be an opportunity to give patients that choice. But that leads me to my next question, which is really pie in the sky, because we're having a conversation about how chemotherapies work. But do you ever imagine a world in which we won't be doing combination therapies with chemotherapy, that we will have first line or even down the line treatments where we can drop chemotherapy altogether because of the long-term side effects and things?
Yeah, you know, we all, in the whole community, advocates as well as clinicians, patients, we want to have a curative agent, or at least an active agent, that has as little side effects as possible. That's ultimately the goal. And we're just still in the early era of immunotherapy. There's newer, more active agents coming along. We're just entering metabolic therapy, and so epigenetic therapy, antibody drug conjugates, CRISPR approaches. You know, there's a lot in the pipeline that may fit that bill. Who knows?
As you said, the future is bright.
Mm-hmm.
Any other questions, comments?
You showed a wonderful plot illustrating the micelle activity in synovial sarcoma, and you said that you obtained 5% complete response rate. Did these patients have something common in their profiles compared to the others?
That's a good question. I don't know the answer to that. That's a very good question.
I think that could be interesting.
Yeah. Okay, well, thank you.
Thank you so much.
Thank you very much. I could challenge you.
Well, I think we can go on we left you later for lunch, but we're giving you time back for the break. How's that? Fair trade-off?